Welcome all. We are going to discuss about the seed. So, scientifically speaking, when you define the seed, what we say that it is the fertilized ovule. That is ovule after fertilization. We call it as seed. That is when the fertilization occurs, that time the ovary where the ovule is present. This ovule is converted into seed, and this ovary wall it is converted into fruit, and remaining parts they wither out. So that fertilization product, or what we call it as post fertilization change, is the fruit. That is, wall of the ovary undergoes some changes. We call it as fruit, and the ovule which is now fertilized, it becomes the seed. So this seed, it contains embryo which is the future plant. It is when we are going to germinate the seed that is going to give us new plant. But in terms of agriculture, if we want to define the seed, there any part of the plant from where we can give rise to new plant. It is able to generate new plant. We call it as seed. That may be tuber, that is root tuber or stem tuber, or even piece of a leaf or shoot, from which we are going to generate new plant. That is, in case of sugarcane, we are using those shoots for plantation purpose. Even in case of potato tubers, we are going to use the buds. That is, small part of the tuber having the axillary bud we are going to plant. So there any part of the plant from which we can generate new plant, we are calling it as seed. But here we are going to talk about the true seed. Scientifically, the fertilized ovule which is forming the seed, about that we are going to discuss over here. So broadly speaking, we classify them into two types as dicot seeds and monocot seeds. Dicotyledony and monocotyledony. The terms itself, they are suggestive that here the two cotyledons are present. It is called as dicot. While the single cotyledonous seeds, we call them as monocot seeds. The general examples, the common pulses or all beans, they are dicotyledons. The familiar examples to you in day-to-day life, they are all pulses. We call them as dicots, or even the beans what I said, they are dicots. So here we have taken typical example of dicot seed, that is castor seed, where we are observing here some external features in the castor seed. The outermost hard region, that is seed coat, which shows the margins which are slightly broader at one end and they are tapering at one end. The tapering end shows presence of irregular whitish mass, that is called as caurucle. It is helpful for absorption of water during germination. Being the hard seed coat, absorption of water is very important for germination. So that caurucle is useful for absorption of that water. Here beneath that you are observing hilum, that is the connection of the ovule with the ovary wall with the help of placenta. So that scar remains, that called as hilum. Inside the whitish flat body you are observing, that is endosperm, which is the reserved food material for maturity of the embryo present inside. So if we take a vertical section passing through this seed, we will find that the outermost layer, which is protective layer, we call it as testa, that is hard one, darker in color. Inside that, the thin layer we are observing, that is the tegumen. The inside layer we call it as tegmen. So testa and tegmen, they are the coverings of the seed. Inside you are observing here two cotyledons. Presence of two cotyledons is there. And at the central axis you are observing the embryo. So in VS also you are observing here the presence of coracle, which is the source of water absorption during germination. So here the embryo axis, that is formed by plumule and radicle. So plumule is going to give rise to shoot system after germination, and radicle is going to form the root system of the plant, that is below ground level. So it is the common structure which we observe in case of dicot seeds. While in case of monocots, the cereals like maize, jawar, rice or wheat, they are forming these monocotyledoni. So here we have taken the typical example of maize, where externally you are observing the structural arrangement in rough sense. What we can say here, the seed is showing one end which is tapering and one end which is broad. The tapering end, it is the attachment of the seed or the ovule with the placenta in the ovary wall is there, and this side it is the style which is attached from the ovary. So this region, it is the broad base of the ovary, where the ovule is present with the basal placentation, and here you are observing that embryo axis which is embedded in the nutritive tissue. We call it as endosperm. This is a food material, that is endosperm. In detail, if we observe this structure in vertical section, we will find the arrangement like this. The outermost layer, that is seed coat plus wall of the fruit which is fused with it, because it is caryopsis type of grain, where the seed coat and fruit wall, they are fused. It is not separated. You cannot separate it by simple ways. Only after grinding, that is hull which you are going to obtain, that is the part of this. Otherwise it is fused. Inside you are observing the nutritive tissue, that is endosperm, which is starchy in nature. By staining it with iodine, you are going to get blue violet stain to this portion, which indicates that it is starchy in nature. Some protein layer is there, that is special layer you are observing, that is hyaluron layer. That is the characteristic feature over here. Towards the tapering end, you are observing the embryo axis. Towards broad end, what you are seeing, deposits, reserve foods. Here it is embryo axis, where it is clear cut differentiation. It is on the upper region you are observing lumen, while toward the lower side you are observing the radicle over here. Here, cap-like protective structure which you are observing on the plumule, that is coleoptile, that is protective layer on the plumule from which shoot system is going to develop. While here on the radicle, similar protective layer is present. We call it as coleorhiza. Rhiza refers to root, and we know that radicle is going to give rise to root system. So protection to the root, that is coleorhiza. So these are the characteristic feature which we observe in these grains, that is in the monocot seed. You can even go for the structural arrangements in the jowar or other cereals where similar kind of arrangement you will get. So it is all about the seeds, that is true seeds, dicot and monocot seeds, from which we are going to obtain the new plant.